In this video, I'm going to walk you through what a viral plaque is in the viral plaque assay. And this works very similarly to the plate count assay or the plate count test. The plaque assay for viruses tells you how many virions or how many active virions there are in a liquid, which is very similar to figuring out how many colony forming units there are in a liquid, which is what the plate count does. But in order to understand this, we need to think about colonies and plaques and what they are. So what I have here, this slanted line is supposed to represent the surface of an agar plate, the surface of the agar in a petri dish. So this is where we would put a cell if we were putting a cell on it to see if it formed a colony. So just imagine, let's be blue. So a little cell falls on the surface of this agar and it sticks because it's tiny, it sticks to everything. So, okay, cool. So what's going to happen? Well, nutrients. This is going to start using whatever nutrients are dissolved in the agar. So remember, agar is mostly water, and agar solidifies the water, and then all the nutrients are dissolved in the water. So down here is where we would have carbon sources, energy sources, electron sources, nitrogen sources, phosphorus, sulfur, all that different stuff in different molecules dissolved in the water. And so as this cell uses some of those, it releases waste products, like gases, like carbon dioxide, and nutrients are going to move towards it through diffusion. So that's going to happen, and that's going to allow the cell to get more nutrients and divide. So it makes it a daughter cell. And then these are both going to divide again, and the cells are going to keep dividing, and they're going to keep dividing, and they're just going to be a whole lot of cells. And eventually there will be so many that we can actually see them, and that's what a bacterial colony is. So a bacterial colony is going to grow, and it's going to keep, it's going to keep requiring these nutrients to move towards it through diffusion, and it's really going to grow until the nutrients can't get to it fast enough, or the waste products accumulate here and poison the cells. So it's going to grow until something goes wrong chemically to stop them from growing any further. So there really is a limit usually to how big a colony is going to get. So this is a microscopic view of a colony. And so we can zoom out a little bit and just remember, you know, if you put a cell down here, it's microscopic, and then it starts to grow on the surface until you can see a colony. And you did it over here too. You can see a colony over here. You can see some here and here. And again, this would be a circular petri dish. If we were looking from above, we'd see a circle, but we are at its level. We are like crouched down looking over the surface of a table, and this is sitting on it. So this is the surface of the agar. These are the colonies on the agar. And they just grow on the surface of the agar because the cells get nutrients which can diffuse through the water to them. It's a really brilliant thing that cells need water, right? Like, well, that's not the brilliant thing, but the realistic thing is that every growing cell is in water or needs water. And this is a way we can give bacteria water without suspending them in water. So they stay in one place. So they make these colonies. Great. So what about viruses? So what about viruses, right? What do they do? Well, remember, a virus needs to grow in a host cell. So if we want to count viruses using a low-tech method in a lab, and this would be one we would do, again, we're going to use agar. Again, we're going to solidify some water with a lot of nutrients in it, but this time we're not feeding the virus. This time we're feeding the host cell. So on the surface of the agar, this time, we'd be feeding this cell. And the cell would have, I don't know, it would have like a nucleus in it and not much else. And at this point, we don't care much about the cell, right? We don't care about the details of the cell. Just know that nutrients are going to diffuse towards it. And so what can happen? Well, a little virus, a little angry virion, the universal enemy of everything, this little virion with its little spike protein sticking out, it can get inside the cell. If it gets inside this cell, what's going to happen? You're going to get new little virions being made until eventually that cell is going to break open and it'll be a dead cell, and there will be the virions kind of fall out and the virions are all over the place. Whoops, that's not, don't buy the kind of graphing pad I bought. That was, yeah, they were hard to find in March 2020. So the little virions will get out. So they basically kill the cell and they get out. So you have to grow a host cell on the agar surface, and it's kind of like that. You grow host cells on the agar surface, and then if they get infected by the virus, they will break open and die. So how do we use this? Well, let's just imagine we have a layer of host cells covering the entire surface of an agar plate. So we've put host cells here, and they have grown slowly, but they have grown all along the surface of the plate. So what happens if we put a virion here? What if we put a virion in this cell? Well, what's going to happen? The cell is going to break open. So for this cell is going to break open, and the cells nearby it are going to break open. And so what we see is like a lighter area on the petri dish where the cells, the host cells, have died and broken open. So it just becomes a clear area where we can basically see through the cells. And so the same thing would happen over here. If we put another virion down, we'd get another area of dead host cells where they are they're breaking open. And so while we can't see a virion and we can't see an individual host cell, what we can see is an area where different host cells have broken open. And we can see it because when one of these cells breaks open, the virions spread to the nearby cells and they infect the other cells nearby. And so we get an area where all the host cells have died, and that is a visible thing. So let me draw that here. So we would look down on the surface of a plate, and we would first, I guess I should write this out, add cells. So we add the host cells. So what do those look like? Those are going to be dark gray and pretty big. So all over the surface of the plate, we're going to put host cells. I did my best fast job of filling in this whole surface with ink or whatever, just like you would do your best to fill a whole surface with cells. And it will never be really perfect unless you get a machine to do it. So you add cells, and then you add your diluted, virions. So your sample with an unknown number of virions, you add to this. And so really then we look at what happens. Well, Wherever a virion falls, you end up with an area you can see through. And we call these plaques. We get a little random pattern of plaques and we count them. And we would do the same calculation we did with a plate count, because we would know our dilution. If this is a 10 to the minus 3 dilution and we get 1, 2, 3, 4, 5, 6, 7, 8 plaques, that's not enough. I forgot, so I'm going to make 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 9, 30, 1, 2, 3, 4, 5, 6, 7, 8, 9, 40. Okay, so let's say we counted 40 plaques. Well, you should pause the video here and tell me how many plaques per milliliter were in the original sample. We're doing the same thing we did before. We're adding 0.1 milliliters of diluted virions. This is the dilution factor, and we counted 40 plaques. So how many virions were in that? Go ahead and pause the video and calculate that, and I will calculate it too. So 40 plaques per 0.1 milliliter times 1 over 10 to the minus 3. So that's like saying 40 times 10 to the 4. So our final answer is 4.0 times 10 to the 5th. And what are our units? Well, this is going to be plaque forming units per milliliter. 4 times 10 to the 5th plaque forming units per milliliter. And so we'd go back to our original sample and we would know there were 40 of whatever forms a plaque, whether that's one virion, it's probably one virion, 40 of them, or 4.0 times 10 to the 5th of them per milliliter. It doesn't tell us how many inactive virions are in there. It doesn't tell us how many dead ones. It just tells us how many things are in there that are capable of forming a plaque. And this is the plaque assay. One last thing about this, because if you followed me to here, that's all you need to know about the plaque assay. It really is like the plate count test, except that instead of growing them, the colonies on agar, you grow plaques in cells that were grown on agar. And then you count them. But here is the key word. The concentration or population size wow. I'm glad my mother is not seeing me write like this. She would be mortified. Population size of virions is called a different word that we haven't used so far. It is called the virus titer, or if you're British, titer. That's an R. And so we would say the virus titer of our unknown sample was 4.0 times 10 to the 5th plaque forming units per milliliter. Okay. Don't be shy about asking me questions if you have questions about this, but this really is something cool we would do in a face-to-face lab. Sometimes we would do plaque assays using bacteriophage because growing human cells in a petri dish is really hard to do, and you need high-tech equipment to prevent contamination that a lot of educational laboratories don't have. So what we would do instead is we would grow bacteria on the surface here and infect them with bacteriophage. And the same thing would happen. All right, cool. Well, thank you for your attention, and I will see you later.